Hey everyone. So it's been a while since I've done a science news article, but the science keeps coming out, keeps getting published, and I found some time to go and look at some articles, and I found some interesting stuff that I wanted to talk about. So first things first, let's start with this really interesting article about air pollution and how it affects the mating capacity of flies. This is this is a topic that might seem familiar to you if you've listened to me talk about how pollutants in waterways affect the sexual development and mating of frogs. This is a very similar issue. We'll read this article, we'll take a look briefly at the paper itself, and then really dig into this and see what's going on. And then there's a whole bunch of other news stories I got lined up for you. So without further ado, let's hop into it. Okay, so this the Max Planck Gesellschaft publication, the article says, air pollution impairs successful mating of flies. High levels of ozone destroy the chemical mating signal of the insects and may thus contribute to global insect decline. Okay, so that's pretty shocking. They're saying, this air pollution, it alters the chemicals, the pheromones that these flies use to mate, and by compromising their ability to mate, it's not a surprise that their populations are plummeting. And we've seen widespread reduction in insect populations across the world of all kinds of insects. So I don't think it'd be too much of a stretch to say that what we're about to learn here about these flies probably also applies to a fair number of other insects as well. Okay, so a research team at the Max Planck Institute for Chemical Ecology in Jena, I don't know how to pronounce that, Germany, demonstrates that increased levels of ozone resulting from anthropogenic air pollution, or stuff that we put into the air, we humans, can degrade insect sex pheromones, which are crucial mating signals, and thus prevent successful reproduction. The oxidizing effect of ozone causes the carbon-carbon double bonds found in the molecules of many insect pheromones to break down. Okay, so there we go. I was right. Many insect pheromones, probably not just flies. Therefore, the specific chemical mating signal is rendered dysfunctional. Most remarkably, the disrupted sexual communication also led to male flies exhibiting unusual mating behavior towards ozonated males of their own species. Interesting. So it not just alters chemical signaling, this sexual communication based on like pheromones and odor, but it's also altering sexual behavior itself. Okay. So this is kind of a big article. I'll try and go through it as quickly as I can, but buckle up. Insect sexual communication relies to a significant extent on pheromones, chemical attractants that specifically allow males and females of a species to mate. Sex pheromones are distinctive to males and females of a species. Even the smallest differences, which we'll see in the study by the way, such as those observed in the formation of new species, ensure that mating no longer takes place, because males and females only find each other through the unmistakable odor of their conspecifics. Most insect pheromones are odor molecules containing carbon-carbon double bonds. Such double bonds are known to be easily destroyed by ozone. So here they start quoting Marcus Naden, who holds the odor-guided behavior group. He heads the odor-guided behavior group in the Department of Evolutionary Neuroethology, or the Neurology of Behavior, at his university. And he's the lead author of this study. He said, quote, "We already knew that environmental pollutants such as ozone and nitric oxide degrade floral scents, making flowers less attractive to their pollinators." That's really interesting. I didn't know that. Since compounds with carbon double bonds are particularly sensitive to ozone degradation, and almost all insect pheromones, sex pheromones, carry such double bonds, we wondered whether air pollution also affects how well insect females and males find and identify each other during mating. Okay, so here it's describing that they created an ozone exposure system for the flies that could mimic ozone levels in the air, as they are nowadays often measured in cities in the summer. So here they're talking about how they set this up. Let's see. First author Nanji Jiang. First author Nanji Jiang said, quote, "We therefore needed a technique that would allow us to measure even tiny amounts of pheromones on individual flies that had either been exposed to ozone or not prior to the measurements. To do this, we used what is known as a thermal desorption unit, coupled to a gas chromatograph or mass spectrometer, which allowed us to measure tiny amounts of odors emitted by individual flies. All right, so yeah, that's super cool stuff. But a lot of the the particular physics of these tools that they're using kind of go over my head a little bit. But okay, so here they say in the experiments, male flies were exposed to slightly elevated ozone concentrations. They measured whether the flies still emitted their pheromone. And then when the flies were exposed to 100 parts per billion of ozone for 2 hours, the measured pheromone levels decreased significantly compared to a control group that had only been exposed to ambient air. In addition to males of the model fly Drosophila melanogaster, the researchers also tested male flies of 8 related species of the genus Drosophila. In only one species, Drosophila buschii, the release of specific male pheromones remained unaffected after ozone exposure. So just 1 out of 8, I believe, weren't affected. But that was only because their pheromone didn't contain any carbon-carbon double bonds, and so they wouldn't react the same way that the other pheromones did with ozone. Okay, now we get into the mating behavior, the differences induced by ozone. The researchers then tested the attractiveness of male flies to their conspecifics. The observations they made were disturbing, which may be mainly due to the role of their respective pheromones. These are emitted by the males in Drosophila species and increase their attractiveness to females. And at the same time, males use the odor to distinguish females from other males. So while their pheromone attracts females, it repels other males. Interesting. So you have that dichotomous opposite interaction there between males and females with the same signal. During mating, males transfer their pheromone to females. Freshly mated females that smell of the pheromone are thus no longer attractive to other males for the next couple of hours. Interesting. So that could have, evolutionarily, that could have consequences for like sperm competition. If you attach that pheromone, if you're a fly and you attach that pheromone signal to the female you just mated with, and then no other male fly is going to identify her as a receptive female for the next couple hours, well, that gives your sperm a few hours head start to fertilize a clutch of eggs. So that's really interesting from an evolutionary perspective. Consequently, elevated ozone levels not only caused females to be less attracted to males, rather, ozonated males were suddenly interesting to their male counterparts. Let's see, who are they quoting here? Okay, both Nanji Jiang and Marcus Naden are summarizing the effects of ozone. They say, quote, "We knew that elevated ozone levels could affect insect mating systems because the breakdown of carbon double bonds, and thus pheromones, by oxidation is not rocket science and chemistry. Nevertheless, we were shocked that even slightly elevated ozone concentrations had such strong effects on fly behavior. Actually, we initially wanted to focus on the interactions between males and females. We could explain that males started courting each other after a short ozone exposure because they obviously could not distinguish ozonated males from females. However, we had not thought about this before. Therefore, we were quite puzzled by the behavior of the ozone-exposed males, which lined up in long courtship chains." The research team also observed the effects of high ozone levels in the air on the mating behavior of other Drosophila species. Even males of the species Drosophila buschii were less successful at mating after exposure to ozone, although ozone does not alter the pheromone that has been described to be emitted by Drosophila buschii males. So that was what we were saying earlier about this particular species has a pheromone that doesn't have a carbon-carbon double bond, so it doesn't have the same interaction with ozone. However, other so far not identified ozone-sensitive chemical compounds may also play an additional role in their mating behavior. In 8 of the other 9 species studied, the research team observed unusual... Okay, so it was out of 9, not out of 8. The research team observed unusual courtship behavior by males towards other males exposed to ozone. Interestingly, one species, Drosophila suzukii, which is known to lack pheromones but courts based on visual cues, was not affected at all by increased ozone levels. So there you go. That's really interesting. By including a species like this, who doesn't even use pheromones and has an entirely separate mating mechanism, in this case visual cues, you can control for potentially other interactions that you may not have thought about at the beginning of your study. So perhaps ozone altered the visual system somehow. Perhaps it affected visual signals, or ozonated males, perhaps it affected like the iridescence or the reflectivity of their carapace, their exoskeleton, or something, that we don't know about ahead of time. But when you include a species like this, you can test that, and you can find, okay, well, they weren't affected, so maybe the visual system is, it's the ozone interference in the visual system. There's no overlap there. So species that mate with visual cues aren't affected by this particular mechanism. And conversely, this also reinforces the fact that ozone altering the pheromones is the mechanism of interest. Like this is just another way to experimentally confirm that there is an interesting interaction going on between the pheromone chemicals themselves and the ozone. Okay, I'm rambling. Let's go on. Okay, so we're almost at the end of the article. They're kind of repeating some of the basic concepts a bit more. Most insect pheromones contain carbon-carbon double bonds. We've said that about 3 times so far. Therefore, ozone is thought to interfere with sexual communication in many insect species. Okay, so they're quoting Bill Hansen, head of the Evolutionary Neuroethology Department and co-founder of the Max Planck Center Next Generation Insect Chemical Ecology unit, which is dedicated to studying the effects of climate change, atmospheric pollution on insects and their chemical... Okay, interesting. How climate change affects insects. So what Mr. Bill Hansen says is, quote, insects and their pheromones have evolved over millions of years. In contrast, the concentration of air pollutants has only increased dramatically since industrialization. The only solution to this dilemma is to immediately reduce pollutants in the atmosphere. There you go, I agree with that. Reducing air pollution benefits everybody, and now it looks like it benefits the animals and the plants too. The scientists and Jenna want to study the effects of ozone on a broader range of insects, including moths that usually follow pheromone plumes over long distances. Sex pheromones are also crucial cues for insects to distinguish between conspecifics and closely related species. So, quote, we would like to find out whether high levels of ozone lead to increased hybridization rates when closely related fly species share their habitat. It's an interesting idea that they just posited here, that if you have these kind of similar chemical pheromones and they're being altered by this ozone, perhaps that might lead to some chemical overlap, or it might alter their interactions in some way that it actually brings these two separate species a bit closer together. That could be the case. Finally, chemical communication in insects is not restricted to mating behavior. All social insects, like bees, ants, and wasps, use chemical cues to identify members of their colony. We also study whether the social structure within ant colonies is affected when ants return from their foraging trips, during which they become exposed to increased levels of pollutants. You don't even want to envision what happens if social structures in ant colonies or beehives suddenly collapse because pheromone communication no longer works. Marcus Naden, who also studies the behavior of desert ants, is concerned. Okay, and then I guess the article here is just ending with general statements about ozone on human health. The current study provides an additional explanation why insect populations are declining dramatically worldwide, apart from the application of insecticides and the elimination of habitats, which, let's not kid ourselves, those are both massive, massive contributors to the loss of insect biodiversity. If chemical communication is disrupted by pollutants in the air, they cannot reproduce at a sufficient rate. This may also affect many pollinators, such as bees and butterflies. The fact that 80% of our crops need to be pollinated by insects makes it clear what scale this problem may take in the future if we do not succeed in drastically reducing air pollution. I mean, if starving to death because your crops fail, because there's no pollinators, because all the insects have died, because air pollution destroyed their ability to mate and communicate with each other, if that's not a strong enough incentive to clean up the air pollution, I don't know what is. A lot of this stuff, at a certain point, it seems like the evidence for this kind of stuff is overwhelming. And the lack of drastic action on behalf of regulators or even the corporations who are made up of people who also live on this planet and eat food, the lack of action is just appalling. But okay, so here is the study in Nature Communications. The article was a bit long, so we don't need to read the whole thing. I'll go over the abstract in this brief little introduction, and then I want to take a look at figure 3 that they have in here, because it's a chart of a lot of the fly species and the chemicals they use and the results that they had when they exposed them to ozone. It's really cool. Okay. Abstract. Insect sexual communication often relies upon sex pheromones. Most insect pheromones, however, contain carbon-carbon double bonds and potentially degrade by oxidation. Here we show that frequently reported increased levels of anthropocenic ozone can oxidize all described male-specific pheromones of Drosophila melanogaster, resulting in reduced amounts of pheromones such as cis-vaccinyl acetate and Z7-trichosine. At the same time, female acceptance of ozone-exposed males is significantly delayed. Interestingly, groups of ozone-exposed males also exhibit significantly increased levels of male-male courtship behavior. When repeating similar experiments with 9 other drosophilid species, we observe pheromone degradation and/or disrupted sex recognition in 8 of them. And I guess with 9 other species, they saw in 8 of them, that 9th species was the one that reproduced with visual cues. And so the ozone exposure the chemical interaction didn't have any effect for them. Our data suggests that anthropocenic levels of ozone can extensively oxidize double bonds in a variety of insect pheromones, thereby leading to deviations in sexual recognition. Okay, so then the introduction, which is just a few paragraphs. Finding and judging a suitable mate is pivotal for reproduction in many animals. In this context, most insects use sex pheromones to discriminate conspecifics from allospecifics and to identify the sex and mating status of a potential mate. A particularly well-investigated pheromone is cis-vaccinyl acetate, or CVA. This compound is produced by male Drosophila melanogaster. It governs sex recognition, and the CVA amount present on a male has been shown to correlate with the male's attractiveness to a female. The effect itself is a product of the concentration of the pheromone. So if they put the pheromone on themselves or they secrete it on themselves, obviously the concentration of the pheromone is going to be highest closer to the male. And so a female who is close to the male is going to get a strong pheromone signal. A female who has been smeared with the male's pheromone to mark her as having mated recently will also have a strong smell, and so will have a relatively high concentration of the pheromone. And males are repelled by that high concentration of pheromone. They want to move away from it to lower concentration of the pheromone, where the scent isn't as strong. And apparently, when the chemical is altered, it smells different. There's a different signal. Males aren't quite sure if they should be repelled or not. And you end up with, on the female side, you end up with lower attraction to the males on average. And on the male side, they appear to get a little confused about which animal's a male, which animal's a female. And so you get these chains or groups of males all mating together. Okay, so they're talking about their pheromones. Although being chemically diverse, most of these potential pheromones share one specific feature. They contain carbon double bonds. And yeah, these double bonds are really vulnerable to being oxidized by a chemical like ozone. During the Anthropocene, insects communicating with such unsaturated pheromones are facing a potential challenge: the oxidation of double bonds by increased levels of oxidant pollutants like ozone. Pheromone systems have evolved in pre-industrial times, with tropospheric ozone values as low as 10 parts per billion. However, due to the continuous emission of nitrogen oxides, volatile organic compounds, and climatic change, the ozone level has already increased to a global yearly average of 40 parts per billion. So what we have now, the levels of ozone we have now, is 4 times what it was in evolutionary history. Local extreme ozone events have been reported for industrial and urban areas, e.g. Mexico, Bangladesh, Morocco, and China. So basically what this means is over industrial areas where there's a lot of factories and a lot of, I guess, just urban development, a lot of combustion, a lot of engines everywhere, heaters, stuff like that, air conditioners. You get these bubbles, or these like heat islands over cities. You also get higher concentrations of ozone as well. And that can affect local populations of insects like these flies. Here we show that even short-term exposure, that's bad, even short-term exposure to ozone levels of 100 parts per billion results in the degradation of many drosophilid pheromones and reduces the attractiveness of males to females in 7 of 10 tested species. Interestingly, ozone exposure dramatically increases male-male courtship behavior, probably due to a lack of sex discrimination when male pheromones become degraded. Exactly. So here they say that a lot of these harmful effects appear to occur at 100 parts per billion concentration of ozone. And so if we look at, I skimmed over this, I should have read this. Ozone reached in Mexico a concentration of up to 210 parts per billion that lasted an hour, and the highest mean value measured over 10 hours exceeded 170 parts per billion. So that is well over the level necessary to cause this kind of chemical interference with the pheromones. Although ozone values vary a lot during the whole year, the yearly measured average for, e.g., northeastern China has increased from 45 parts per billion in 2003 to 62 parts per billion in 2015, and reached a daily maximum average for 8 hours of 140 parts per billion observed in March 2020. Okay, so you can see the trend there in like the northeast China plain, where China produces a lot of its food and where a ton of people live, like 400 million people live up here. It reached a daily maximum average over 8 hours that was well over the dangerous threshold that they observed in the study. So that's disturbing and awful. Okay, so if you scroll through the study, you can read some of the technical specifics about what they did. I've already been talking about this for a while, so I'm not going to go through all these technical details. But here, figure 3, I wanted to take a look at this graph, or this chart. So here they have the species. Here's a scale, 1 millimeter, so these are about like 2 to 3, maybe 4 millimeters long. Drosophila melanogaster, the control species. I think that's what CS means. So here is their chemical, that CVA. And then you can see Drosophila putrida also uses the same chemical, as does Drosophila repletoides, although they also use a new one called Z11 to 20 AC. But yeah, this is just interesting, looking at the different chemicals that have been evolved. You see these two groups, they use the same one, Z10 to 17, 2AC. These guys use a similar one that's slightly modified. Or this species, Drosophila buschii, they use two different ones that are kind of closely related. Might be the result of like a genetic replication event. Really interesting stuff. And then if we look at this column, these little graphs here, you can see the normalized intensity or the concentration of the pheromones in normal air versus ozonated air. And you can see in the ozonated air, the concentration of the pheromone is generally less. Its intensity is less. In groups like this one, like Drosophila saltans and Drosophila Moj wriglei, I'm not sure what Moj stands for. You see it's significant, like really significant. And then if we look at these columns, these bar graphs here, we have mating success and likelihood of male-on-male courtship. So mating success, you can see that there is slightly less, significantly so in these species, less mating success when there's been ozone exposure. And then here, the courtship, these results are really quite stark, honestly. You can see that the courtship behavior between males, so this male-male sexual behavior, skyrockets when they've been exposed to ozone in almost all the species. Like it's significant in D. melanogaster, D. putrida, Reploides, Funibris, definitely significant in Drosophila pallidipennis, pallidipennis and Drosophila saltans. Interestingly, here in these two related species, Wiglii and Mohavanensis, it looks like exposure to ozone reduces male-on-male courtship. Interesting. But then you see elevated levels of it again in Drosophila buschii and non-significant elevated levels in Drosophila suzuki. So all told, altogether, a super, super interesting study. A little terrifying, a little concerning, because, insects are important. Their ability to communicate and mate with each other and sustain their populations and pollinate plants is important. So the fact that air pollution disrupts that and puts a lot of stuff at risk, stuff that's downstream of insect populations, like plants that need to be pollinated, including our own agricultural crop plants that we eat to sustain our entire civilization. Big red flag, guys. This is important stuff. Might want to clean up the air pollution sooner rather than later.